Genetic variation and what its origin and how we detect it. Genetic variation refers to difference in DNA sequence between individuals of same species. This variation is the fuel for evolution, enabling population to adapt to changing environments and giving rise to the incredible diversity of life we see today. Origin of genetic variation: mutation. Changed DNA sequence can occur spontaneously during DNA replication or through exposure to environmental factors like radiation or chemical. This mutation can be small changes like single base substitution, or large change like insertion or deletion of DNA segments. Recombination. Another method, recombination during sexual reproduction. Homologous chromosomes, one from each parent, exchange genetic material during a process called crossing over. This shuffles genes and creates new combination of allele, further increasing variation. Gene flow, the movement of gene between population through migration or breeding, can introduce new allele, increase variation in the recipient population. Detection of gene variation. Cytogenetic technique. This method involves visualized chromosome under microscope, allowing for detection of large-scale chromosomal abnormalities like duplication or deletion. DNA hybridization technique like Southern blotting, DNA microarray, use labeled probe to identify specific DNA sequence, enabling the detection of mutation or polymorphism. Different version of the gene. DNA sequencing. Direct sequencing of DNA molecule is the most comprehensive method for detecting all types of genetic variation, including single nucleotide polymorphism, insertion, deletion, and copy number variation. The study of genetic variation has numerous applications. Understanding the evolution and adaptation. By studying how genetic variation change over time, scientists can trace the evolutionary history of species, understand how they respond to environmental pressure, improving agriculture, identifying genes associated with desirable traits in crops and livestock, can help develop new varieties with increased yield, disease resistant, and other beneficial characteristics. Medical genetic. By analyzing genetic variation in individuals' different diseases, researchers can identify gene mutation involved in disease development, paving the way for personalized medicine and the development of new